Good afternoon, everybody. We are really happy to start this series of scientific webinars here at St. Peter's School. Today's webinar is about biotechnology and genome editing. And our keynote speaker is Dr. Pareras. Dr. Pareras is the Director of Health Equity, a venture capital fund investing in healthcare. Dr. Pareras is a neurosurgeon, holds a PhD and an executive MBA as well. He's also the author of more than 10 books in the field of innovation, technology, venture capital, and healthcare. And without any further delay, I will let him start with his presentation. Go ahead, Dr. Pareras.
Thank you so much, Lourdes and Teresa, first of all, because and congratulations to the school for making this happen. I think it says a lot about your school, these webinars and this keeping updated in science. I think it's very important. And what a wonderful opportunity to talk about science as well. To talk about the science means to talk about the truth. In this era of the fake news, etc., I think we all have a responsibility to be more science focused and more data driven. So congratulations for this webinar. First of all, I want to tell you, first of all, that I really envy you, all the students present in the room, because literally you're gonna live amazing things in your life. This is only starting. What I will present today you'll see several things that are quite interesting, but this is only the start of it. So a revolution is coming. And I really envy you. Also, I envy you because you're the first generation that probably is going to live for more than 100 years in average, each of you. And this is something astonishing, and you should feel really privileged about the world that you're living in, despite all the things that are going on, etc. Right? So the most important thing that I want you to understand in this lecture is that life is programmable. Okay, this is the assumption behind all gene therapy, gene editing, and synthetic biology. And today's class is going to be about synthetic biology, precisely, CRISPR and synthetic biology. So this is the underlying concept. Could you please project the first slide? I don't know if it's, yes, Teresa is on it. Okay, so what happened in the last 70 years in medicine that has been so extraordinary? Just 70 years ago, and I'm sure you know about that, I'm sure Corinne has already explained a lot about these issues, but in 1953, Watson and Crick discovered the DNA structure. They first described the way that life passes on the information to the next generation. Just a little bit after them discovering this DNA structure, the medical community started finding out that a lot of diseases were genome related, were diseases because there were mistakes on the DNA. Okay, in fact, more than 7,000 diseases are related to DNA distortions or malfunctions or errors in the code. And then it opened the field to 3 successive fields that are really redefining what medicine can do. And this is truly a revolution, okay? I'm gonna try to describe, just to conceptualize what each field does, just for you to understand. I know that you have done this already with Corinne, but I find it very useful to try to understand exactly what that means, okay? So first, we had gene therapy. And gene therapy is just the transfer of genetic material to a patient. Let's imagine, for example, the whole genome, your genome, okay? It has roughly about 35,000 genes. Imagine like a library with 35,000 books, okay? So gene therapy would be the equivalent of taking out one book, every book is a gene, okay? One book that is badly written, that has some problem in it, and then replacing it with another book, okay? This is This is exactly what gene therapy does. In fact, what it does is we don't take away the book, we just add a new one that has the correct instructions for the protein that this gene produces, and then the protein is produced inside the body, and then everybody's happy because things start to work again. But this is very cumbersome because, as you can imagine, a gene is long, and then you need to change the whole book. And then someone invented, and we're going to talk about that a little bit later, someone invented a way of edit the genome, not just changing one gene with another, but getting inside the gene and change the letters that are wrong. It would be the equivalent of a word editor. It's like using Microsoft Word, but for a gene, okay? So now with the library analogy, we're not just taking out one book and replacing it with another. We're just opening the book. We're finding the sentence that is wrong, because it's usually a very short sentence. Sometimes 30% of the time it's just one letter that is wrong. Okay, so it's a base nucleotide that it's wrong, and we need to change it. Sometimes it's a sentence, okay, but we go inside the gene, in this case the book, and we change the sentence, okay? So the mutation is corrected in situ, in the place that is wrong. But we weren't happy with that, and then we are starting now what's going to be the most important revolution of the 21st century, which is going to be synthetic biology. It is not just that we correct now a gene or edit the sentence. Now we are programming a new book. We're introducing a new book inside the library just to make the book do things that were unthinkable before. Okay, so now we are programming life. We are, in a way as well, creating life, because as you will see in a few slides, we can create very basic forms of life already, but we are starting to be able to create life. And that's indeed the future of medicine. Could you project the next slide, please? So this is synthetic biology precisely, and we could use this programming life thing just to precisely program new, let me just move this for a second. Yes. Now, we could use this, for example, we can program cells to do something. In this case, we could program cells to produce something, okay? So we will have some cells, we will introduce the gene, the new gene that codifies, for example, for the production of fuel or of light or of new garments, and then have the cells produce something. So we could use biology to program cells to produce objects for us or tissues for us. The cell as a living factory, okay? So large-scale factories. This is being done. This is not science fiction. This is already being done. We can create, for example, artificial silk. We can create garments, for example, with cells that change their color depending on the light, just as chameleons do in real life, because the gene is exactly the same one. Imagine, for example, a coat in the future that could change color. This is being done as we speak today. We have, for example, cells that we program them to emit light, so we could have light bulbs, but not normal light bulbs, but living light bulbs, etc. So we are producing consumer goods. In this case, life is the product. Okay, but we don't need to stop here. We could also use the cells to introduce them inside humans to solve some disease. Imagine, for example, this is being done very extensively. Let me introduce for a second what I do for a living, just for you to understand this. I mean, I am a venture capital investor. That means that I invest in emerging technologies in life sciences. Okay, so I invest in many companies. We just invested, for example, in a synthetic biology company that tries to provide a vaccine for COVID-19. We'll talk about that a little bit later. But these emerging fields I mean, what I do for a living is precisely receive maybe 400, 500 ideas every year, and I say no to 495, you can understand that I'm the bad guy probably, because I'm the guy that says no. But I invest in 5 of them, and these 5 I try them to reach the marketplace. And I have plenty of examples of these things that I'm telling you, of companies that are willing to get to the marketplace doing precisely this. For example, in healthcare, we could program T cells to fight tumors, for example. And this is being extensively done through CAR-T technology. Maybe you have heard about it, chimeric antigen receptors technology. We can program cells to fight against infections. We could program cells in the field of immunology. We can program cells to detect toxins, for example, inside the body and fight against those toxins, etc., etc. Okay. Then the third field that we are starting as well in synthetic biology is to release on the environment these cells to do something. For example, in bioremediation, this is very useful. Imagine a cell that is programmed to eat all the plastic in the ocean, metabolize it, and provide with inert metabolites that are not harmful for the ocean. The same with pollution, the same with biomining, a very interesting field, just to extract metals in a much more productive way. Food production, you have all heard this artificial meat revolution that's coming on. This is done precisely with synthetic biology tools and technologies. We could even use DNA in this case to store, as if it were a hard drive, okay, to store information. Of course, it has a problem that to access the data and to record the data is not very fast at the moment, but a lot of people are working in the world very hard to make it really, because the great advantage is that it would last a lot of time, and every DNA molecule can hold a lot of information, you know. So this is very interesting. So when programming life, we could use this programming cells concept to have the cells produce something, to introduce them in human beings to solve something, or to release them in the environment to do something. Could you project the next slide, please? And here we have, just for you to know, this is the paper that changed everything. You know, this is the paper that was published in 1953 by Watson and Crick, the molecular structure of nucleic acids. I'm just introducing it in the lecture because I want you to really go to the source always. When you hear something related to science, don't just stay with what the speaker, in this case myself, says. You should go to the source and check and read it, okay? So this is the original paper. Could you project the next slide? And this is exactly how CRISPR works. And I wanted just to give you a few concepts, because I know Corinne as well has explained to you, but CRISPR is a construct made by 2 parts. It's the single guide RNA, which is the part of the CRISPR system that locates the place where we want to do the edit, okay? And then the Cas9 nucleases, which is a protein that is designed as a scissor. I mean, it makes the cut exactly what the guide RNA tells the Cas9 to do the cut. Okay, there are many types of Cas. This is a more complex slide, and we won't get into biological details, but many different types of Cas. The Cas9 is a more usual one, but then you have the CPF1, which is the high fidelity Cas, also known as Cas12. You have dead Cas, which means that you can interfere with the gene without changing it. We have Cas9 nicases, which just produce one, I mean, not a double strand break, but one strand break. There are many variants to do that. But in essence, what happens is what you can see at the right of the slide, you know. The place of the cut is detected by the guide RNA, and one thing that's called the PAM that's in the original DNA. Then the guide RNA binds to the DNA, then the Cas9 undergoes a conformational change and becomes the scissor that we want, and then it produces the double strand break. This DSB, double strand break, is essential because from that moment on, the cell tries to repair, because the cell is programmed by millions of years of nature evolution to repair that cell. Then you could have, for example, the non-homologous chain joining, which would mean that the both extremes of the cut are repaired immediately. But if you provide a template of the gene that you want to edit, sometimes you get this homology-directed repair, which is the other mechanism the genome has to repair, that introduces this strand of DNA in the place where it's needed just to make the correction. This means that in any gene therapy, or in any gene editing in this case, you will have always cells that have underwent the correction and cells that haven't, but sometimes with just as few percentage of cells as 5% of the cells corrected, that means that the disease disappears, but they start producing the protein and then the disease goes away. I didn't want to get very technical with this, but just to let you know that CRISPR-Cas9 is precisely these two parts of the molecule. And one very important thing, this guide RNA has two parts: the red one, which is called the spacer, or CRISPR RNA, CRRNA, and then the scaffold, which binds to the Cas protein. Okay, this spacer is just 20 nucleotides long, and just with these 20 nucleotides, we are able to find exactly the place where we want to do this cut. Of course, if these 20 nucleotides are repeated elsewhere in the genome, we could have a CRISPR cut in some other place that's not our intended place to cut, and this is called an off-target effect. And this is the major problem that CRISPR-Cas systems have right now, but we are trying really hard to solve that problem just to make it even more precise. It is already very precise, don't misunderstand me, but we can make it even more precise. Could you project the next one, please? Class, I give you here the picture of the first living thing that escaped the evolutionary tree. Maybe I'm going to be very, this is going to be very hard for you to learn, but Darwin is dead. There's no longer, once this happened, Darwinism is dead because we are controlling the evolution. This is a picture of something that Craig Venter did just 10 years ago, which is the creation of a cell from scratch. This cell was constructed with programmed genes, with a minimum set of genes that make a cell viable. Only 400 genes make a cell viable. If you take away one gene of this cell doesn't live. So we got it precisely for the right amount of genes that make that thing not only live, but grow, eat, and divide. Okay, so this is an artificial cell that was constructed just programming with the 4 letters of the DNA code. We just use the adenine, thymine, cytosine, guanine just to build that cell. And once the information was constructed, this was the output. Could you project the next one? But yeah, but we are not even limited by this. I want you to understand that until very recently, the boundaries of biology were set by nature. But now these boundaries are set by scientists and human beings, because this is the first paper published with a synthetic DNA that was using not just the 4 original letters, but an extra 2 letters. This is what I like to call the first alien being on Earth, because this is not the result of evolution. This was precisely created with new programming code. Okay, this paper is amazing. I really recommend you to read it. Of course, Teresa and Lourdes, this set of slides you can share with all the students, but please find this paper because it's amazing how they did it and the astonishing results that they got precisely. Okay, could you project the next slide, please? Yes, and this is my final slide, and I want for you to think right now what we should do. And I'm going to ask you, all of you, through the chat to let me know what's your opinion, and I accept all opinions, and all opinions are individual opinions, and that's the way it should be, okay? But listen, in this 2x2 matrix, I try to summarize what we can do today with genome editing. Okay, and this is a moral dilemma. Okay, this is an ethical issue. We could use genome editing for therapy things, meaning to correct diseases, or we could use editing to enhance the human being. And at the same time, we could do editing just on somatic cells, meaning that this editing that we do doesn't get to our descendants, to our sons and daughters, and to their sons and daughters. Or we could do editing in the germline, exactly as what happened, for example, in China with this crazy scientist that already has done CRISPR intervention in 2 kids that were born and are alive. These 2 kids have their germline affected. Okay, so let's do it that way. There will be 4 letters: A, B, C, and D. Okay, A is the option where you are only comfortable with correcting diseases and in somatic cells. That means the green area that you have here in the slide. Okay, that would be A. B would be doing therapy not only in somatic cells, but in the germline as well. Meaning that if we're going to correct a rare disease, let's do it forever, not just for this generation, but let's remove the genes for all their descendants. Okay. Option C is doing editing as well for enhancement, but only in somatic cells. That means, for example, we could improve humans now, better individuals. For example, I don't know, making them see a bit better, or we could add, for example, a 6th finger in their hands. We know the gene of polydactylia. Where is it in the genome? We could add a new gene to have 6 fingers, whatever, you know. And then option D would be the full spectrum, would be doing enhancement as well on the germlines, which means precisely that Darwin is dead, rewriting evolution. Okay. And this, of course, is not being done now, but would you be comfortable in the human race doing that, controlling its own destiny by rewriting evolution's rules? So I would love you to vote, each one of you, which of the letters would you feel comfortable with? I don't know if I will see the results because I'm doing this from a phone, but I would love someone from the teachers to tell me what the results are. Not the exact results, but more or less which is the option that is winning.
So Joan, Mauri, maybe you can monitor the chat. Joan?
At the moment, B was winning, but it's quite difficult to control it now. I could say,
of course.
Most of them, they say B. Some of them, they say A.
Okay.
Yeah.
Anyone saying D?
Yeah, some of them they say B. Yeah.
Very interesting. Okay. Let me tell you my opinion now, but this is everything that I explained until now. It's science. Okay. Now I'm going to express an opinion, which is not the truth. It's just my personal opinion. Okay. Because there are debates, there are very long debates about this issue in medicine right now. Okay. My opinion, my way of thinking is always safety first. Safety first means we can only perform the changes where we know exactly what we're doing. We can't experiment at this point doing very crazy things. So at this point, I'm comfortable with A. Okay. And I would even be comfortable in a few years, once we know a little bit more, with B as well. Because I think a rare disease, if it's of genetic origin, we should be able to change it once and forever, you know. So there's nothing wrong with correcting a disease forever. The problem is, are we causing any harm in future kids precisely by doing that? And as we don't know, I prefer to be prudent. But let me tell you something. In China, they're doing B and C like crazy, okay? And some of their experiments are not known at this point and will never be known because they are performing this. In fact, CRISPR is so easy to perform that it could be done in a garage, literally. So this is coming. For example, anyone thinking we should enhance, well, why not? Plastic surgeons have been enhancing for more than 50 years, or ophthalmologists have been enhancing the human race for a lot of time. So there's nothing wrong with that in itself. The problem is, are we causing harm? And if you follow the principle of safety first, then maybe it's a bit too early to do that. And finally, this rewriting evolution, I mean, rest assured, this is gonna be done. This is gonna be done by someone with or without permission. And this red circle that I introduced in the slide means the things that are being done right now. There's a little bit of enhancement, and we can conceptualize why sometimes we need to enhance things, because, for example, if a disease is progressing, we could aim a little bit higher just to let the disease progress, okay? And of course, it has been done in China, in the germline, which I think it's crazy at this point, but it has been done. But rest assured, it will, I mean, this circle will expand over time. And what we accept as society depends on what we consider as normal. You know, what is normal? Maybe normality, the concept of normality will shift in this century, you know, because what was normal maybe 20 years ago, what wasn't normal 20 years ago, maybe today it is normal. And then maybe 20 years from now, it will be normal. So let's not assume things. This is an ongoing debate. Society is going to debate it quite hard in the coming years. But what's for sure is that this is a revolution. This never happened in human history. The ability to program life, to make life a product, and this is going to change things. Finally, I'm going to just to end, I'm going to tell you, I bet you that the Amazons, Teslas, and Apples of the future, of 10 years' time, are going to be synthetic biology companies. And you heard it here first, okay? But rest assured, they're going to be these kind of companies that are moving forward very quickly. And now I think it's my 25 minutes already, and I'm ready to take any question. I'll be more than happy to answer if I know the answer, because first and foremost, what I am is a scientist, okay? And scientists, we only deal with the truth or what the things that we know. If we don't know them, we say we don't know them, okay?
I think in order to go for the questions, I would say, I think all the students can write, they can type request for questions in the chat, and then we will give them the turn to talk, just to make it organized.
Excellent. I see the chat. The problem is, again, I'm doing this from a phone, and I'm not daring to change my screen right now. Maybe you could select, Joan, the most interesting ones, or maybe Corinne, I don't know, and read them out loud for me.
Okay, yeah, it's not a problem.
Quería decir que algunas de las preguntas que nuestros alumnos querían hacerte están contestadas. Entonces, yo les he dado un poco un orden para que fueran preguntando lo que quisieran o lo que tenían apuntado, y si os parece bien, puede empezar la primera persona, que me parece que es Dulce. Dulce, me parece que eras la número 1. A ver quién me ha dicho esto. Pero la de Dulce está contestada, ¿vale? Entonces, pues la persona que viene después de Dulce, a ver, un momentito, perdón, podría ser Lucía, ¿no? Es Lucía. Si quieres, Lucía, empezar tú con tu pregunta. Y luego, pues, podéis ir siguiendo el orden o interviniendo, como queráis.
Sí, yo, Corín, también quiero preguntar luego.
No. Yo, más que nada, era un poco para darle como un orden, pero aquí todo el mundo pregunta.
Pues pasamos el turno a Lucía, ¿no?
Vale, Lucía, ¿quieres empezar tú, Lucía Sanzoné?
Puedes activar el micro, Lucía, y lo puedes hacer directamente con la voz.
Sí, yo preguntaba si tenía alguna relación esta técnica con la de los transgénicos.
Ujú. Vale, sí, sí tiene que ver, evidentemente. Mira, un transgen, justamente, vamos a ver de dónde viene la palabra. Transgen, sorry, I'm skipping to English just in case you're recording this and maybe this is going to be used afterwards, but a transgene is just a gene, a new gene that has been introduced remotely into the genome somewhere. Okay, so yes, the answer is yes. Every transgene is a cell or a species or a product that has been manipulated using gene editing techniques or gene therapy techniques. So yes, these foods, for example, that are artificially in a way, which by the way have been demonstrated across many studies that are absolutely safe. They are exactly the same as normal food. Use gene editing technologies to be produced. That's correct.
Muchas gracias.
De nada, Lucía.
Ruth, si quieres continuar.
Next question.
Corín, ¿quién va ahora?
Ruth Maldonado. Supongo que está.
Vale.
No
Sí.
sé si mi nombre o no. Yo tenía curiosidad por saber si en este ambiente científico de desarrollos y todo esto es un ambiente muy competitivo o predomina más la colaboración entre científicos. Gracias.
Wow, what a great question, Ruth. This is a very important question, very interesting question. Listen, it's very difficult to answer because among scientists, when you consider only science, there's a lot of cooperation, but there's a lot of competition as well, because all good scientists want to publish first in Nature or in Science or in the Lancet or the New England Journal of Medicine their findings. Okay, so it is ultra competitive, but it is, I would say, a very healthy competition because they compete, but scientists are very trained to admit and to quote everything that they learn from elsewhere, from other guy or woman, and I would say it's a very intense competition, but a very healthy one as well, because they are very fair. It's not like companies competing. You know, what I see when I go out is people competing really hard, but in a very fair way, just pretending to be the best at what they do. And I think that's great. That's what moved forward science. Yes. By the way, in venture capital, we definitely compete because we invest in companies, and therefore, once we have invested, we compete. But I'm a scientist first. Before being a venture capitalist, I was a neurosurgeon. In fact, I still am a neurosurgeon, but I now work in the venture capital field, investing in companies. And yes, there's a fierce competition. Fierce competition. Great question, by the way.
Vale. La pregunta de Nicolò creo que ya está contestada. Era sobre si se podía construir una célula con los genes que quisiéramos y está contestada,
Yes.
me ha dicho. Entonces, quizás es Darío Prada, que tendría la siguiente pregunta. Darío, what's your question?
Well, I would like to ask you if you know, or if you think that there will be a vaccine for COVID-19, and when?
Super interesting. Could you project my last slide, Teresa? Listen, this is, again, a very important question, and I can give you full certainty on my answer because the correct answer is I don't know. Okay, there are viruses that we have tried long time to get a vaccine, for example, HIV, the AIDS virus, and we never succeeded. So there are viruses which is very difficult, or I would say even impossible to get a vaccine. Other viruses are very easy to tame and to have a vaccine. This is not my last one, I think. This is one that says SARS-CoV-2 vaccine, because I was expecting this kind of question, you know. And this, no, This one, this is the one, exactly. No, the previous one, this is the one, exactly. Here you have all these strategies that we are trying to use to build a vaccine for SARS-CoV-2, okay? And except the first one, which I call the classical approaches, all the rest are based on synthetic biology. I can tell you which one is going to succeed. My personal bet would be number 6, RNA vaccines. There are several. Moderna, for example, is a very powerful company in the US that's trying to bring forward an mRNA vaccine encoding the spike protein on the surface of the virus, which is the best place probably to try to teach our immune system to detect and find and destroy the virus. But there are more than 90 projects ongoing, 5 of them already in phase 1 in clinical trials. Phase 1 means testing their safety in real human beings, not in animal models, but in humans, okay? I think we need a vaccine very fast, because otherwise, without a vaccine, this is, I mean, I prefer not to be, I prefer to be on the upright side today, but this is going to be more difficult than what you read on the media. Everybody wants a vaccine as fast as possible. Maybe we're going to beat our record in the time that we need to produce a vaccine, but I think it will never be before next year, okay? But in any case, it could happen. And if someone comes up with a good strategy for a vaccine, then excellent. If it's this year, I would be very surprised, but I don't think that's going to happen. In fact, I don't think it's if we will ever get a successful vaccine. Maybe we'll get some sort of flu vaccine-like, which, as you know, are vaccines that are not very efficacious. They provide you with 70% of protection, but 70% of protection is not that high, isn't it? So the answer is, I don't know. I think we will, but I think it will take at least 1 year from now. What I can tell you is more than 90 groups are working extremely hard in the world to produce one. And I have my own experience because I've invested in a company that probably will start production for a hypothetical vaccine using synthetic biology techniques in the coming weeks. Okay. Next question.
Next question sería Aina Canosa. ¿Tenías tú la pregunta siguiente?
Sí. Well, hello. My question was also related with COVID-19, and is that I wanted to know why it affects so much to some people and almost doesn't affect to others. Thank you.
Wow. If I knew that answer, Aina, I mean, that again is a very good question. The problem with COVID is that it produces what we call a cytokine storm. It's what more technically it's called a cytokine release syndrome, which means that your own immune system overreacts in the presence of the virus, and it overreacts especially in the lungs. Okay, so people get a respiratory distress and they need a ventilator, a mechanical ventilation, because otherwise they die. That's why so many people have died. And it affects young people, old people, people of all ages. And we don't know yet, Aina, why that happens. And I can assure you, we will know. We will know precisely why this happens. But so far, nobody has identified the common characteristic of those people that get affected. Probably it's something related to a genetic predisposition, or it's something related, I don't know. I can't assume things because the correct answer is we don't know, but we will find it. But I wish we knew, because this would mean a lot for the control of the pandemics.
Anna Q.
Hello. So my question is also very related to the vaccines and COVID-19. And I was just curious, why is it so difficult to get a vaccine for COVID-19 while every year we get a vaccine for different flu viruses?
Yes. Yes, it is very difficult for a number of reasons. A lot of reasons could make that this vaccine is difficult. Among them, for example, let's use HIV, the AIDS disease, right? HIV is very difficult to vaccinate against because the very same virus mutates inside the body almost immediately after infection. So every single individual has many different types of AIDS, HIV viruses inside himself. And the virus mutates very fast, and it's very difficult to have something that really provides a very good vaccine. Also because the AIDS virus also attacks precisely T cells and the immune system, so it makes it even more difficult to produce a vaccine. It could be, for example, because the virus mutates too quickly, but I don't think that's going to be the case in COVID because already it is already 4 or 5 months since it was discovered. By the way, its genome was published just 15 days after being discovered, so it was a significant success again of synthetic biology and our tools. And this could be one of the reasons. Maybe this virus will be, I mean, maybe we will obtain a vaccine in 1 year and there won't be any problem, and it could happen. But also there's something that worries me because normal infection, there is a doubt, and this is very serious, about whether if the infection provides with good immunity after having been infected, you know. Normally you get an infection, your body develops an immune response to that infection, and then you get immunity at least for 1 year, normally for 5 years or even more, against, sometimes even for life, against that infection precisely. But if the virus mutates, then you lose all you had won precisely because of being immune. But in this case, in the COVID, there are significant doubts that it provides a very good immunity after having gone through the disease, and this is very bad news. If we confirm that, which again, we don't know yet, but the first results that we're getting are not very encouraging, okay? And I'm really worried. This is the number one thing that worries me about COVID. It's not the seroprevalence, it's not the arrival of a vaccine, it's not whether we should confine or not confine. My number one concern is precisely your question. Does it provide good immunity after infection? Because if it doesn't provide good immunity, that could be another reason why we won't find a vaccine very easily. Because if even the same virus, the virus itself doesn't provide immunity, how come will we be able to provide for that immunity? You understand? So it's tricky, but again, let's hope we will find one. I'm confident eventually we will find one. Not quickly, but we will.
Thank you.
You're welcome, Ana. Very good question.
Simar, creo que puedes preguntar.
Yes, so basically my question is in regard to the Cas9 protein or any Cas protein from its family. Does it only come from bacteria or like prokaryotic cells, or can like eukaryotic cells like animal or plant cells also synthesize this type of protein? Thank you.
Very good and very technical question, Simar. This is a very interesting question because, yes, it has been studied whether, I mean, because the origin of Cas systems, of course, is in bacteria. This is a defense mechanism of bacteria against viruses, okay? But we're using these Cas systems into humans. We have discovered several Cas systems that can be used in humans, but some of the humans have already developed some immune reactions. It's what we call immunogenicity. It means that Cas9 can provoke an immune reaction on the host where it's used, okay? And this is bad news because if that's the case, then we should have several, I mean, if we want to perform several times CRISPR-Cas9 editing, we should be very aware that this could cause problems in the future for human beings, you know. But every time that this happens in the past, when some hurdle is there that prevents us from doing something repeatedly or anything else, we find a solution for that. So I'm not worried about that. I think we will come up with a solution, because you could use even synthetic Cas9s, which I guess that's the answer that you were looking after. We could use synthetic Cas9s that could be very useful, and that could, for example, maybe not trigger this immune response. Okay?
I see. Thank you.
You're welcome.
Vale. Stacy creo que tiene preparada la siguiente pregunta.
Yeah, in the future, could this technique be used to fight viral diseases?
To what, sorry? To?
In the future, could this technique, be used to fight viral diseases? Like,
of course it can. And in fact, it is already being tested as well. This is not a vaccine, this is a treatment then. We're talking about a treatment. But CRISPR-Cas9 can be used against the genome of the virus. Of course it can. And I'm betting you we will come up with something to treat this virus using these techniques. Again, this is a matter of time. Of course, we need to find something that works as quickly as possible. And I'm pretty sure that the fastest response won't come from a CRISPR-Cas9 attack on the virus. But I think eventually somebody will use it to fight against infections. In fact, editing is used against many infections in proof of concept in animal models right now as we speak. So yes, the answer is yes.
Thank you.
You're welcome.
La verdad es que hay 3 o 4 preguntas más que ya han sido contestadas durante la charla. Entonces creo que la última que tenemos nos la va a hacer Aina González, porque las demás están todas contestadas. Aina, cuando quieras.
Hello. My question is, if in the future CRISPR is used for option D, so for somatic cells diseases enhancement, could there be a case of social injustice because of the cost of it?
Wow, what a last question. This is the most tricky and most difficult one to answer. I think there's an issue there, because you are very right. If we use, of course, CRISPR would be used first and foremost in the first world, okay? So that in itself shows a bias, because it won't be used first in the third world, so there would be some kind of social injustice. But even in society, in a specific country, we could find cases of social injustice, because as everything in life, it is a tool that gives you the power to do things or to solve things, and it would be expensive. So the social systems of the world are going to counterattack that precisely by trying to pay for that CRISPR out of the money from the governments, you know, as it usually happens. But even in that case, there will be some sort of social injustice. But let me tell you something. Even if it creates a little bit of social injustice, these social injustices happen only for a period of time at first. But then once the technology is well established, it gets to everyone. So I think we shouldn't be fighting that. Although I understand the question and I see your concern and I admire your concern, I think we should go ahead and still perform all those experiments, even if at first we cause a little bit of social injustice. Because many people that are low in the scale, that couldn't afford that, would get benefited because the state will pay for that as well. So I think we should go on and do it, even in that case. Thank you.
Okay, thank you.
Joan, Mauri, em sembla que te hemos pasado todos por delante, ¿no?
Sí, ningún problema.
Ahora Joan me hace la pregunta que me desmonta por completo porque
No, tranquilo, no, creo que no. So yeah, let's talk in English if you want, just to finish the. Okay, I cannot stop thinking about all the possibilities. I still remember your schematic about A, B, C, and D, and I think it's limitless. And I would like to know if you know the limits, because now I'm thinking on my field of interest, which is technology. And when you say program, I think about software. We can program the software. It's something that we cannot touch, but it's modifying, and it's giving us an outcome. And then we have in the robotics field, we have the hardware. Okay, so my question is, will we be able in the future to modify our software and our hardware as a humans?
Absolutely.
Is this included in?
Yeah, absolutely, yes. The answer is a radical yes. We will be able. You're asking if we will be able. Should we do it? That's a very different question that goes into the ethical realm, and it's very difficult to answer. But this is a problem now. I mean, if you come from the software field, this is very similar with what is going to happen with artificial intelligence. Should we do it? Shouldn't we do it? What are the limits? Is it dangerous? Is it not? I don't know, because software, hardware, yes. But what worries me is artificial intelligence and genome editing, precisely because they transform the human race and our society in such a profound way that the changes, once they are performed, they are very difficult to take away, you know? So, but I'm very optimistic. Of course, science is a tool, as technology is a tool. It can be used for the good, it can be used for the bad. It just amplifies, you know? Is genome editing good or bad? Well, I don't know. It's an amplification of what we can do, and you can use it to amplify the good, and you can use it to amplify the bad. So it's dangerous, yes. But I'm optimistic because I think human beings, there are lots of crazy people around, but at the end of the day, the majority of us are solid mentally, and we are not crazy. And I think we will take the necessary measures to control the fallout of everything and the spread of these technologies. Because one thing is for sure, we shouldn't allow this directly without limits and without regulation. This can go out without regulation. You know, we need to control it, and we need usually to go a little bit slower than what people want. But I'm for going slower, and I'm for doing things very carefully, but doing things after all, okay?
Okay, thank you so much. I'm so excited. So I hope to program a cell or something like that soon.
Super. Excellent. I don't know if there's, yeah.
Corinne, do we have more questions, or anybody else has more questions?
Me cuesta seguir los pensamientos en inglés. Si es ciencia, me cuesta menos. No sé si entra dentro de lo que has preguntado, Mauri, pero yo digo, nosotros los profesores, ¿cómo podemos preparar a nuestros alumnos para este mundo que les viene, no sé, atravesado por la ciencia por todas partes, con los problemas éticos? ¿Qué debemos hacer los profesores para preparar a nuestros alumnos?
Gran pregunta, Corinne. Yo creo que el mundo que se nos viene encima, es decir, el mundo del siglo XX, por ejemplo, era un mundo donde lo que se enseñaban eran cosas, datos, etcétera, ¿no? Es un poco, yo creo que el mundo del siglo XXI lo que hay que enseñar es precisamente enseñar a aprender cosas rápido en un entorno muy cambiante. O sea, no enseñar los datos o las cosas, sino enseñar cómo aprender esas cosas en un entorno muy cambiante y dar las herramientas a los estudiantes pues para que, sobre todo, sean hipercríticos, que sean librepensadores. El mundo del siglo XXI es un mundo de librepensadores, gente que opine por sí misma y no se deje influir por los demás, ¿no? Que estén comprometidos con la verdad y no con las fake news. El mundo está derivando hacia las fake news de manera terrible, en mi opinión, y tenemos que concienciarnos todos. Yo creo que la ciencia y los profesores, sobre todo vosotros, los científicos también, los profesores tenéis que enseñar mucho a distinguir, a que la gente sepa buscar las fuentes, sepa leer los artículos, sepa cuestionar y dudar de la palabra de los demás y solamente aceptar lo que venga de referencias en las que confíe o lo que uno mismo pueda haber entendido, etcétera. O sea que yo creo que ahora la educación tiene que estar más fundamentada, desde luego no en datos, no en las antiguas listas de los Reyes Godos, por supuestísimo, sino en estimular la curiosidad. Y me dirás, ¿y cómo se estimula la curiosidad? Bueno, tú eres la experta, yo no. Pero te digo, es lo más importante, ¿eh? Yo creo que la curiosidad es lo que mueve a la gente y lo que la hace apasionarse por las cosas. Y yo creo que si hubiera una manera de estimular, de enseñar curiosidad, que no sé si es posible, sería my number one choice.
Muchas gracias.
Si me permitís, y para no abusar ya de Luis, que además creo que nos hemos pasado de tiempo, te lo agradecemos, me gustaría solo hacer una reflexión. La charla que hemos tenido ha empezado muy científica y ha acabado absolutamente humanística. Desde mi punto de vista, en los colegios lo que tenemos que hacer es justamente, yo creo que lo que ha pasado ahora mismo, que es a través de un medio tecnológico, en unas circunstancias absolutamente adversas, ha venido un experto con artículos en la mano y ha sabido enlazar a la perfección los valores éticos, humanos y a la vez la innovación científica más puntera de hoy en día. Entonces, yo dejaré terminar a Teresa, pero quería agradecerle pues esta inspiración que nos ha provocado hoy. Y estoy convencida, yo soy de letras, o sea, yo enseño lenguas, estoy convencida que hoy habrá surgido más de una vocación científica. Muchas gracias.
Gracias a vosotros, Lourdes. Y es un comentario muy, muy pertinente, porque además yo creo mucho, o sea, la ciencia es el punto de partida, pero el humanismo y el arte tienen que tener un peso muy importante para construir no solo mentes, sino personas, ¿no? Y estoy muy de acuerdo contigo que la ciencia sin humanismo y la ciencia aislada de todo, sin arte, sin sensibilidad, no es nada. Por lo tanto, creo que es la combinación de todo.
Okay, we had one last question, Pat, because she's asking for a little time to ask another question if we can.
Of course.
What do you think? Yeah. Okay, so Pat, go ahead. Pat.
Yeah. Can you hear
there
me?
you go. Oh,
Yeah,
yeah,
okay.
sure.
Right. I have a question. I want to go back to those kids, those twins in China whose gene pool was edited, if I remember correctly, to protect them from the HIV virus. And I was wondering, I also heard that the doctor who did this experiment has had his license revoked. But I was wondering if there's any follow-up in terms of physical health, emotional health of these twins. I'm assuming now they're probably maybe 2, 3, 4 years old, and if there's any transparency and any findings about how they're doing.
Very good question. So far, I mean, of course, as you can imagine, these kids are being really monitored across time since their childbirth, which is in itself such a burden for them, you know, because they will eventually know that they were edited before birth. So far, nothing has come up, but we can't be sure it won't come up in the future. And I just need to remember you about the Dolly Mutton oveja. I don't know if you're aware of the Dolly experiments. Yeah. When they created an animal, and after, I mean, it seemed normal, but their lifespan, the lifespan of these ovejas was really shorter than the normal lifespan. So we need to be very careful about that. I'm not saying this is going to happen to these 2 kids, of course not, and I hope it won't happen, but they are being very closely monitored. Let me tell you something. This doctor in China was accused of doing things very badly, and he faced a death penalty. After that, he was commuted, and he has been, revoked of his license to do things, and things have not gone so far. But he was facing a death sentence. So, because China is as it is, I think what's going on in China with these new technologies is very dangerous, because they are not used to regulation. They are not used to local authorities just regulating the things that can and can't be done. And this provokes scientists to do many more things than the things that we are doing here. And without knowing what are we doing, again, I'm going back to my first principle, safety first. This is very dangerous. So if you are very powerful, if you have the power to change the genome, this is a very big power, and every big power demands a big responsibility as well. So the thing that he did was outrageous. I totally disagree with his view on doing that, and I hope it won't be repeated. But again, this is gonna, I mean, this will happen again and again and again. So we need to make sure that we are very, very aware of the situation and of the problem, and we need to try to take some measures to at least make it so difficult for it to happen that we diminished a lot the times that this will happen. Okay.
Thank you.
You're welcome.
Okay, so the questions keep coming. I mean, we still have a few more questions, but I know we are a little bit maybe behind time.
No, but
So,
I'm okay,
no?
because
Is it
so
okay?
yeah, if you want maybe a couple more, and then yes, at 6:30 I have another conference call, but I already scheduled just yeah.
Okay.
Two more, it's
we
okay.
have yeah, two more. So Nuria, you seem to have a question. If you want to go ahead.
The microphone, Nuria.
Or maybe we can wait. You, Mauri, had another one, no? Joan?
Yes, so while we wait,
Okay.
I watched the
Oh, she's
documentary.
here.
Okay.
Nuria, if you want to go ahead, go
Yeah.
ahead.
Yeah.
No, I was just thinking about after what Lourdes said, because she was actually reflecting on what being human means, and then how humanities are needed actually to make like efficient science, because we need to be creative, but that also means rethinking what actually being a human is and how we think, and then connecting it to what Mauri was saying. So Mauri was asking, I'm trying to get the two so that you see the thread. So Mauri was asking, will we be able to edit the software and the hardware? And I'm thinking, when we change the DNA, when we edit the DNA, theoretically, I would say we're changing probably the hardware. But then how does that change the wiring that we've got inside and the software of how do we behave? And so, I don't know, I'm trying to think of the implications of this field also will change the way in which we are humans and sciences and social sciences, I guess.
Absolutely, the way we think, in a way, isn't it? And my fair answer to you, I mean, this is again a very important question, but I don't have an answer for you because I don't think anyone has one at this point. We don't know how this will change us, but of course it will. And it will change ourselves, and it will change social structures and the way we behave, and even our ethics and our morality. Because imagine a world where I don't know, you could make copies of people just to have spare organs in case of a transplant, or I don't know. There's so many things that we could do that they are very efficient, but so not human in the way that you are talking about humanism and about being human beings and caring about each other, etc. That it's a bit scary, to be honest. So things, I mean, our software is going to change when we change the hardware, but we don't know the implications of that. That's precisely why we need to do this very, very slowly. We are not in a hurry. We need to be aware of the consequences before doing this mainstream. Yeah.
However, I'm going to take it now to the other field, and I'm thinking that somehow we as teachers try to change that software without editing the hardware. Yeah. So
absolutely.
we try to change that from other work. So that's also it also leads us to reflecting on what's the role of the environment and the experiences that we are undergoing.
Very interesting. And let me come back to you as well by saying that then you have a very important responsibility for the future, precisely because you are tampering with this software, and you should, I mean, we should use all the tools that we have to change our hardware without changing our software, or if we change our software, changing it for the good, you know, to be better human beings and to have more fair societies, etc. Yeah, and that's, maybe you are at the forefront of that responsibility, so good luck.
With all technology though so far. Okay, thank you.
Do we have any other questions? We have time for one more.
Okay, yeah. So I don't know if you know the documentary called Unnatural Selection. It's it's on Netflix, and they talk about biohacking.
Yes.
And it's quite interesting.
I haven't seen it, but I will after this question, of course. Natural selection.
Okay,
but
I really recommend it because everything that you have explained today, yeah, it's really related. And you talk about curiosity, and I think it's really important, but also it's important to balance the curiosity, because in this documentary they go deep into this topic, and they also go to these 4 different parts, A, B, C, D, and they argue about the D point, and the scientists say that our ecosystems are already broken, and we have to go to the D point because it's the only choice we have. Okay?
Interesting.
And in addition to this, they explain that there is a kit online that you can buy, and you can modify a transgenic cell. And actually, it's not really expensive.
No, as I was mentioning, precisely this can be done in a garage at this point, because it's very, very cheap. It costs some $100, something like that, or euros. And you don't need very expensive equipment. So it can be done in a garage. And in fact, there are examples of people that have auto-injected CRISPR modifications to themselves, you know. One of them died, you know. This is not a game, and we should be very aware. Of course, this curiosity, I have an example for this curiosity thing, that too much curiosity is not interesting, you know. But it's about precisely the way of guiding the curiosity. For example, if one of our kids is playing in front of, I don't know, something that's hot but not very hot, you know, you want him to experiment, to touch, and then when he feels the hot, he says, wow, this hurts, and this is useful. This kind of curiosity is useful. But for the same token, if our kid is playing with a gun, of course you will go very quickly and take the gun out of him, and it's curiosity as well, you know. There's some curiosity that's dangerous. That in that I agree, but of course, as everything in life, it depends on the consequences, you know, of the action and the curiosity, where it could drive you. The problem with human beings is that we are very curious where artificial intelligence will get us, where synthetic biology and genome editing will get us, and but we should go slowly again. My message today is, we are not, I mean, lots of things could be done, but maybe shouldn't be done, or at least shouldn't be done right now. We need to learn a bit more and do it in a careful and humane way.
Okay, thank you so much.
You're welcome.
Okay, I think that was it, all questions. At this point, we should give you a big round of applause. I don't know if that's possible, because everybody has the yay. I think it's.
Thank you so much. That's my first ever applause on Netflix.
Yeah.
Never happened before.
So well, thank you for the webinar. And as I was listening what you were saying, I thought it was at the beginning a webinar of things that will happen in the future, but they are already happening now in the present. And as a teacher, I think we've learned a lot from this webinar. And I hope all our students, they learn a lot as well. And as Lourdes said, maybe some, you know, vocations were sparked here for the future. So just to say thank you, and
let me say as well,
webinar number 2 in the future.
Super. It's been really a pleasure. Again, congratulations, Lourdes and Teresa and everyone at the school, because I think that says a lot of what kind of school you are, and again, teaching science and truth and debate and humanities and moral debates and issues. It's very important. And I would love all students, regardless if they go to the science branch or they go for more humanities and everything, to really be aware of what truth is, and to try always not to accept exactly what people say, but to think for themselves. It's what we said about being free thinkers, okay? And this is very important. And anyone in the science branch that's interested in this type of slides that I presented, I've got a Twitter where I publish these slides. There are hundreds of them. And if you're interested, just find me at Pareras in Twitter, and we could follow the conversation there as well. So thank you. This was an amazing experience. It was really a privilege for me to talk to you today, guys.